Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back to the Genetics Podcast, everyone. I'm excited to be here today with Leeland Ekstrom, who's the CEO and co-founder of Nashville Bio, goes by NashBio for short, which is a for-profit subsidiary of Vanderbilt University Medical Center. If you haven't come across them before, I've been following the company for a long time, and Leeland and I actually met years ago at this fun, interesting Illumina event that we can talk about if we want to. I think probably the first and only such event. But since then, Leeland has built an amazing company that is doing something pretty unique at combining genetics with deep clinical data within the Vanderbilt University ecosystem. So Leeland, welcome. And I'd love if actually you could start with the origin story of NashBio, because it's a bit of an interesting one. You were started as a consultant there, but actually find yourself as the CEO and co-founder now many years later since you started that consulting project, probably not knowing quite where it would lead.
Yeah, it is an interesting story, and it's not something I could have predicted, certainly when I started my consulting career or even partway through. I'd worked for Vanderbilt Medical Center a couple times on some different consulting work, and particularly around commercializing innovation and genomics, personalized medicine had long been a focus of the medical center. So a lot of the work we had done for them was in that general domain. With the original creation of the BioView resource, and this kind of predates my interactions with Vanderbilt by about at least 10 years or so. They were finding they were having a lot of inquiries from outside entities wishing to make use of that resource in terms of their own R&D activities, primarily coming from commercial entities, pharma diagnostics companies, asking how they could make use of the incredible data set that Vanderbilt had assembled in the form of BioView. And so after some different trials, I guess I'll say, they took the typical sponsored research approach with having different labs facilitate different investigatory products. And I think the conclusion they reached eventually was like to truly take advantage of what they had built and really maximize the potential. They decided to build a business in front of it. And so they retained us to help write a business plan around that idea. So how do you take an incredible data asset and then transform it into something that actually has commercial potential? And so we ended up writing a business plan over the course of a few months. I was both the lead consultant and the lead client. I had taken a leave of absence from my consulting role at that point, and so was directing the consulting team, but then kind of firmly entrenched on the Vanderbilt side as well to both make it more easy to interact with folks at Vanderbilt, but then also to start to assess the market from a business development standpoint, and much easier to do sitting on that side of the house. And so we got to the end of that initial business planning effort, and the leadership of the medical center basically asked me, how much do you believe in the business plan that you've just written? You've spent all this time and effort.
Right?
Can you go build it? And so that hadn't been part of the original plan. The original plan was to come and write the business plan and go back to my merry consulting way. And obviously the challenge of executing what you've done. So we started more earnest business development activities at that point. Started developing some initial test points of the business plan we'd written. Really, it was applications of different statistical genetics techniques. Some of our colleagues at the medical center had really done a fair bit of work around drug repurposing and trying to use early versions of Josh Denny's PHOS methodologies to find new uses for existing or generic drugs. And so those are some of the first commercial areas that we started developing, as trying to help with some portfolio prioritization analyses using PHOS to try and identify new indications for early stage targets. We developed that into a couple initial projects. But for me, the real hook was when I started hiring people to join. And that was kind of what snared me at that point, that it would have been really hard after I've convinced a couple of people to come on board and start doing this, to all of a sudden bail out and say, okay, good luck guys. Yeah, it was about an 18-month process there, from kind of initial business plan to when we had things really operating. We were in a bit of a stealth mode at that point. We were inside the medical center while we were sorting out some of the incorporation and legal entity side of it. So we operated with inside Vanderbilt for probably the first year and a half time frame. And then early 2018 is when we formally launched NashBio.
And it's not been that long, actually, 2018, but you've accomplished a lot in that time. One of the probably really big milestones recently was completing sequencing of a whole genome sequencing of 250,000 individuals. I wonder if you could talk about that, because it feels like one of these resources that is probably already paying a lot of dividends from a drug target discovery prioritization perspective. But as you and the clients you work with start to think about how you link this with other similar data sets around the world, like UK Biobank, that's at 500,000 whole genome sequences now, not quite the same clinical data footprint as you have. Wonder if you could talk about the origin of that resource a little bit, because it's one of the very few, I think, at the scale that we're talking about, right, where you have not just the genomic data, but also really deep clinical data across, in this case, a geographically constrained population.
Yeah, as I said in my prior comments, I can't take credit for any of the architecture of the underlying resource in that regard, really standing on the backs of some visionary folks at Vanderbilt that first started this whole thing in 2004, 2005 timeframe and enabled all of that, which has come afterwards. As we were in the early days of NashBio, we expanded beyond some of those initial use cases around drug repurposing, and quickly realized that the ability to build very exquisite and very specialized cohorts using the clinical data we have access to. Just as a short digression, we have that de-identified extraction of Vanderbilt Medical Center's electronic health records. So it's a very rich phenotypic resource in that regard. We have kind of all the structured components in terms of labs and diagnoses and codes, but we also have all the unstructured components to it too. So things like clinical text and imaging and waveforms and kind of all the color and richness that really comes in the healthcare interactions. And so we quickly learned that kind of the ability to combine all those different elements and build a very powerful cohorts, whether it's longitudinal or looking at drug responders or progressors or kind of all those things, and kind of expanding into the building of those different cohorts. So in the early days of NashBio, we were doing that on a kind of a bits and pieces basis. We'd work with somebody in a specific disease area to build a cohort in kidney disease or liver disease or something like that. We'd request the samples from the biobank and go and sequence, whether it was an exome or a genome, but it was all kind of little bits and pieces. And obviously then just the logistics of having to wait for samples to come out of the fridge and go off to the lab and sequence. I mean, there's a 3 to 6 month turnaround as well. So just the speed at which we were able to operate was limiting. And so probably around 2019 timeframe, one of my co-founder and our chairman at the time had come up with the idea. He called it sequence the fridge, so to speak, that we had all these samples sitting in the fridge. We were pulling them out in little bits and pieces. And the goal would be, all right, well, what if we took an entirely different approach? And this was shortly after, I think certainly the UK Biobank Exome Consortium had just been announced maybe the year prior. And I think the whole Genome Consortium had just been maybe announced a few months prior to that. So we came up with this idea. It was just before the pandemic. I think we'd been in Nashville for a board meeting, and I think I had to go home to help out with the kids right before the holidays. And he went and had pitched the idea to some of our contacts at the medical center. Obviously, everybody loved it as a general thing, and the track had been laid down by the UK Biobank, but I don't think any of us kind of appreciated what was involved in trying to pull off an initiative such as that. So fast forward a couple months, we were out starting to canvass and solicit interest, and that's right when the pandemic hit. So this would have been spring of 2020. There was still a lot of interest, but I think just a lot of uncertainty at that point in terms of how R&D budgets were going to play out in different pharma companies. So that did slow us down for a few months, but kind of pushed through it. We quickly found with a lot of people working from home, and there was even more demand in terms of accessing data sets of the type that we had. So we went through a few different iterations, and this is the stuff that kind of happens behind the scenes. We talked to some different partners or potential partners. We ultimately got connected to the folks at Illumina. One of the leaders at the medical center was connected to a senior person at Illumina from a board they were both sitting on. And that's where, in that milieu, the idea of joining forces with Illumina came together to move this thing forward. And so from there, it was a couple different tracks. One, we had to figure out the relationship between us and Illumina, but more importantly is continue to solicit interest. And that was the thing I don't think any of us really anticipated the challenge of getting a number of large pharma companies, even if you've got an attractive asset and a compelling value proposition in mind, getting a number of large companies together. So we went through a whole bunch of different pivots and transitions over the course of probably about a 2 or 3 year time period. So from 2020 to 2023, when we first announced the sequencing, all kinds of zigs and zags as we weaved our way through that. Ultimately, what we found worked most successfully was getting a couple of the members really interested and then kind of bring along others, as opposed to try to get all 8 lined up at the same point around the same vision. But what we found worked best was getting a couple champions, so to speak,
Right.
in a couple of the companies and using them to bring their friends along. We had some additions and subtractions, so we thought we had one, and then somebody would step away. But ultimately, like bringing a core group together, and we started out with the announcement in January 2023 with the folks at Decode, the Amgen subsidiary, who agreed to do both the sequencing and services of the lab, but also take the kind of the first leap and commit to sequencing the first cohorts. I started off with 35,000 individuals of majority or suspected majority African ancestry, and then 6 months later announced the addition of 4 more members: Merck, AstraZeneca, Bayer, and AbbVie coming on, and then over the course of 2023 picked up a few more. So BMS, GSK, and Novo Nordisk to close out the alliance late in 23. So yeah, eight large members on board. The basic premise was to whole genome sequence 250,000 samples from the biobank fridge, make that available to the members to help power their R&D activities, and then flow that data back to the research community at Vanderbilt as well as a way to build the resource. And so it worked out. So far, it's worked out pretty well. From an operational standpoint, we literally just sequenced the last sample last week, and so we're working on kind of the final data release. We'll have more to say about that shortly when we'll call it done. And then starting to think about what comes next from an expansion standpoint. That interestingly, there was nothing magical about the 250,000 number. That was kind of what the total biospecimen count that we had available, and we started, and we've accrued another 100,000 over the course of that period of time. And then obviously watching with interest at some of the other directions the big resources are going. So certainly proteomics is another direction, but just the move into multiomics in general. And so we're trying to think through what's the most logical extension for us in that regard.
Yeah, so along that thread of the initial, identifying partners, negotiation, that sort of thing, I would imagine from where I sit that you probably get a lot of different reactions. There may be some pharma companies that just say, we're all in on genomics, and it's a core part of the way we want to discover drugs and stratify patients. And so UK Biobank is great, we're in it. Our Future Health is great, we're in it. We want more. Let's figure out how to do this here. But there's probably some other people that are saying, we're already in UK Biobank. Is this incremental? Is it different? And there may be some people who say we're not part of UK Biobank for whatever historical reason. And so maybe this is our opportunity to be in early with a resource like this that could help us. How much of those different archetypes do you see? And are there some that dominate where there are particular companies that are the leaders and they're all in? And how does that play out? Because you probably get that, and you'll get that again with proteomics, right? Is this incremental to UK Biobank, or is it's a different population? It's the US. It's probably a different ancestry and ethnicity backgrounds. So I'm just curious to hear about the kind of different trade-offs people think through there.
It's interesting. We didn't compare notes on that before we spoke, but I think you probably described the 3 archetypes pretty well that we saw. There's definitely champions or kind of the leaders in the field, those who've made the large investments in genomics as kind of their core strategy. And Regeneron's an example. They're not in the AGD per se, but have led a number of the other consortiums. Decode's obviously a leader in that regard. There's a couple others that were clearly like all data. More data is better. We'll take all data. And so certainly hitching or getting a couple of those groups involved and interested right from the outset was a key piece of it. I think the second group was certainly we've missed out to some degree in a recognition that data is an important part of the strategy. And we, for whatever reason, historically, we didn't join UK Biobank or FinGen or any of those others. And so this is a way to catch up in terms of getting a big bolus of unique data. And I mean, we have the advantage of it. It's a more constrained resource or more, I guess, contained resource in that regard. So UK Biobank, as you said, it's a tremendous resource, but it's available to the world. And so you could argue that you have to work a bit harder to find unique things at this point from a resource that everybody has access to. And that was certainly part of the pitch to those groups that hadn't necessarily been involved in these types of activities. That here's a way to get kind of turbocharge your data acquisition strategy. And then the third group was probably somewhere in the middle. They'd been involved in some of these, and again, just looking to either catch up or continue to expand. Maybe they're not interested in the whole data set, but they're interested in certain populations, and take layered on top of all those archetypes. And maybe this third group especially was groups that we'd worked with in some fashion prior to the creation of the alliance. So we'd studied a few different disease areas, and in the interest of moving the alliance forward, we tried to make it as attractive as possible in that regard. So yeah, if you were interested in multiple cohorts, we tried to make it such that it made economic sense to join AGD in that regard. Yeah, and so that was certainly the third group. But from a kind of separate from the archetype standpoint, I mean, how do we differentiate it from some of the other initiatives that are out there? And certainly depth of clinical phenotype was one of the key drivers that we tried to highlight, that going back to that electronic health record, EHR from one integrated system, so it's all normalized and coming from the same source. And yeah, you can debate one site versus multi-site, and there's pros and cons to both applications. We certainly leaned into the pros of having kind of one consistent set of practices and just fitting the data all together. But that depth of information, both the structured and the unstructured components, were certainly a big part of the pitch to the members that did join. The longitudinal aspects of the data set are an important part, and in my prior comments, we've seen that more and more as over the course of NashBio, that people are building more complicated phenotypes and really trying to look at dynamic aspects of it. Why are patients progressing? What is the speed of progression, or who's maybe not progressing? And is that a sign of a possible therapeutic avenue? And responder, non-responder. So kind of all those components require that depth of information, as well as a data set that moves over time. You can't really do that with just static, simple case control definitions. You really need to be able to track patients over time. Certainly diversity of the demographics was another key point for the members that came in. So things like, we cover all different disease areas in the BioView resource. There's no exclusions from a disease standpoint. Vanderbilt's a major tertiary academic medical center, so they have every major specialty and a broad base of disease. And so for somebody looking to get access to the whole resource and maybe has a large pharma with a number of different therapeutic areas of interest, you can kind of cover all those bases in a single resource in that regard. But age diversity was another component of it. The resource covers pediatrics all the way up to geriatrics. And so that was another key differentiator from a couple of other resources that are more population or targeted specific age demographics. And then ancestry was certainly another piece of it as well. And so with that, the first about 12% of the data set sequenced was that individuals of diverse ancestry. We were trying to select individuals thought to be majority African ancestry. Obviously, until we ran the sequencing, we couldn't verify that, but we did some things to try and enrich that first cohort. And it's as far as we're aware, it's the largest whole genome sequencing cohort of diverse ancestry at this point. I know there's other initiatives underway that will eventually extend that, but it's a real paucity of data, even at this point, in terms of being able to study non-European individuals. And so thinking about different diseases that exist in different ancestral populations, this resource enables that. But also even just basic understanding of the genome and how the genome's evolved over time. They're starting to bring in some of those more diverse ancestries. I know certainly a couple members, that was a key point as well. Just the basic science questions, in addition to the more translational stuff that's going to come out from studying disease, or the ability to study disease outside of majority European ancestral populations.
Yeah, interesting. I was kind of going to ask around the rough rank ordering of different motivations. My guess a priori would be that new target discovery, novel target discovery is probably top of the list. Is this non-responders market positioning, understanding, can we develop something that might treat people who don't respond to first and second line therapies? Is that a second place, distant second, close second to the target discovery, and they're kind of related in some sense, right?
Yeah, I mean, I think those are almost subsets of the same activity, right? Is like whether you're looking for a modifier target or some kind of complicating factor that would then inform the basics of it. Most straightforward in Mendelian, kind of true Mendelian conditions where you're looking for modifiers, but I suspect that framework holds in other disease area and basis of complex disease, right? It's like multigenic or polygenic in that regard. So if target discovery is the overall application, I think simple phenotyping and more complex phenotyping, it's how you go about using those data to try and define ultimately what you're doing. I think in terms of moving downstream, though, looking at protocol development would certainly be another aspect of it. How can you eventually recruit trials to find these people? That when thinking about how you run a clinical trial, if you've got a genetic component in that protocol, you want to minimize the genotyping or sequencing that you're doing in terms of that screening. So there are clinical characteristics you can pluck out of a population to kind of increase your enrichment in that regard. We're a de-identified resource in that regard, so you can't use it for trial recruitment explicitly, but you can certainly squeeze as much out of it as possible in terms of trying to understand how to better design a trial in that regard. Admittedly, genomics has not moved too much further down, I think it's still in the early days in terms of moving further downstream. So we've dabbled a little bit with pharmacovigilance activities. We did some early days in NashBio, we did some drug safety predictions, and I think there's a vein there, but it's not in I wouldn't say that's mainline pharmacovigilance practice at this point. And then thinking about true commercial applications, yes, rare disease companies are certainly trying to hunt for patients and kind of that patient journeys and trying to understand how patients are moving through a healthcare system. That's maybe another application. But back to the AGD, the members that did join, I suspect target discovery and ultimately pooling this resource with other large data sets they've gotten as a way to boost statistical power in their analyses. That was the key motivator, and all these other things are going to come along as they have access to the data set and play with it over the next years.
How have you thought about the product and the technology that sits on top of this? Because it's striking me as a major challenge that you have to deal with, which is at the base layer, you've got sequencing pipelines, ingesting all the clinical data. That in and of itself is a beast. You've got to make that available to the sponsor clients that you work with in some kind of trusted research environment. But even on top of that, like all these questions we're talking about, pharmacovigilance, feasibility for trials, target discovery, you probably have choices to make of, do we just allow them to bring their bioinformatics and computational biology teams in there and do their own thing? Or do we start to abstract that away and say, hey, we have built a patient journey mapper in the Nashville ecosystem that means we can export things?
Yeah.
How do you think about like where you just let the computational people go nuts and do their thing, and where you think about trying to build products?
Yeah, and I think that's been a big part of the NashBio evolution over time. In the early days, we were working very heavily with the professional services component and bidding for somebody from a consulting background. But I mean, it was our fingers on keys. We were building very specific resources or very specific data sets kind of to spec from the companies we were working with. We'd then provide that data in whatever format. We've always been cloud-based in that regards, whether it was putting it into a cloud bucket that they can access or whether it's transferring to them. But it was kind of a heavy layer of our teams in that regard. And I'm still trying to figure out if it's these big pop gen initiatives that have pushed this, but I'm seeing more and more interest in kind of these all-you-can-eat types of activities where companies have direct access to the data sets themselves.
Yeah.
So in parallel to the AGD model, we are in process of working to deliver something that delivers that to clients who aren't in the AGD. So being able to access our whole data set and then be able to play with it directly. So standing up our own TRE that we can then open up to clients. I don't think that's sufficient I think it's necessary, but not sufficient to succeed in the space. And kind of the mantra we've adopted is the combination of data, technology, and services. And so data by itself is where we've operated historically. Data plus services is the layer we've had on, and now bringing on the technology component where we will stand up a TRE. But just having the technology, the data by itself, I don't think works either. There's a number of companies out there that for various reasons are still going to need help. And some of that is just understanding clinical context or how to work with these types of data. Real world data for practitioners who are not familiar there's some challenges, and you need to know what you're doing. And oftentimes the early research folks that are doing target discovery are not necessarily fluent in some of those clinical aspects. Those people sit in other parts of a large pharma company. So having that facility with real world data, but then also the clinical context of how disease decision making gets made. And so examples of that would be presence of ICD coding, for example. ICD coding is a billing system we use here in the US. It's how hospitals and insurance companies talk. And yes, it's informative, but just because somebody has an ICD code doesn't mean that they automatically have that disease. And so understanding some of those nuances of how the delivery side of healthcare works, and therefore when you're using these types of data, being able to correct or adjust for some of those effects. And so that's where kind of building the more complex phenotypes come in. And that's where the services component is still essential. So you said kind of the foundation of genomics is the baseline. We actually think the clinical data is the baseline upon which all the pieces build. And so working on in that TRE, having access to the full structured clinical data model, and then having kind of modular add-ons that sit on top of that. So if somebody wants to study cardiovascular disease, you have that baseline, and then you'll be able to add a cardiovascular enhancement on top of that, where we've gone through and extracted a number of the interesting features out of kind of the cardiovascular relevant parts of the health record. So maybe to describe that more specifically, we went recently and climbed through all the cardiovascular notes, whether it's echocardiograms, electrocardiograms, kind of all those components that are used in the diagnosing of cardiovascular disease. Rather than expecting our clients to be able to climb through those data types, we've done those extractions and then put those into a cardiovascular specific add-on that can be then built on top of that. And so moving to an architecture where you've got the generalized base, and then being able to add these data products on top of that add context and color in specific therapeutic areas. And then genomics becomes one of those add-ons as well, because there certainly are a bunch of use cases that just make use of the clinical data, and then genomics comes on for certain aspects of it, and then over time adding other omics layers to that resource. So self-serve model for those who are able, navigated model for those who need the extra help, but in all cases trying to do as much as we can of moving data from a raw state to a more derived or refined state, so that those the companies we work with don't have to mess around with the munging and the cleaning to be able to do that. They can take it and work on the science they're interested in, not in the data jockeying.
Vanderbilt is a major academic medical center, but it's not unique in that regard. You've got Mass General, Mayo Clinic, Johns Hopkins, Cleveland Clinic, UCSF. Different places have different versions of this, but I think you all are really significantly built up compared to almost all the others. And the question I have is, if someone were thinking about, suppose we wanted to do this across every academic medical center in the U.S. and also nation states, right? We have this discussion in the UK where I'm based of, do we have a national treasure that we're sitting on? And is there much more that we can be doing? Do you think that some of the learnings and some of the technology could be abstracted away into some of these other contexts, or is there so much baked into the specifics of the center that you're at that it needs to be built on a center by center basis to really work?
Yeah, I mean, we've thought about that a fair bit, and I mean, as I've noted a couple of times, and we had some particular advantages coming into this. That 20 years, Vanderbilt first adopted electronic health records 25 plus years ago, which was probably a decade sooner than a lot of health systems were doing that. The biobanking of samples, obviously, that just takes physical time to accrue those samples.
Not everyone has that, right? They didn't start 20 years ago.
That's right, and you can't make up for the opportunity or the lost opportunity in that regard. That said, I don't think others can certainly replicate that. And we've already started to see others coming to market in that regard, who whether they borrowed the architecture from Vanderbilt, or Truvette is a good example where they're going to embark shortly on building a large scale resource. But I think in all cases, it is that scale point, right? Is that there's more and more interest in larger these resources as the costs of sequencing and data generation have gone down. It is feasible to start talking about millions or eventually tens of millions of subjects in that regard. And the reality is that's going to require us to figure out how to knit together these different resources in different places. There's no one health system, whether even a national system, like NHS, right? It's still distributed across a bunch of healthcare is inherently local in that regard. So we're going to have to wrestle with this challenge here in the US. There's some unique challenges of each. Yes, there's networks of health systems they're knitted together, but even electronic health records within a given health system differ across those resources or across those different sites of care. So we are going to have to figure out how to start stitch these things together. I do think we are well positioned to help catalyze some of that. I mean, I don't think the solution here is 50 NashBios with every health system creating its own sort of NashBio. I do think we play an incredibly important layer in terms of that translation from the health system to the commercial world. And so I often come back to a mining or resource analogy where all these health systems have mines or raw ore sitting in the ground, but they still need somebody to dig that raw ore out, refine it, make it useful, put it in formats, and put it in places that companies can ultimately pick it up and use it. And so one of the things we're thinking through right now is how can we help catalyze that type of activity. With the announcements of the Alliance, we've heard from a number of AGD, we heard from a number of other data sources around the world of like, how can we join forces or how can we participate with you in some fashion? So we're working through a prioritized pipeline in that regard. Could we start to bring some of these data sets together where you're putting them in at least the same data model and vocabulary, and ideally starting to normalize some of the clinical context? And bringing one of the major points we heard from our customers is NashBio as a data provider, you're well positioned to be able to do that type of stitching together. Obviously, there's complexity in terms of how you do that, whether you're dealing with, even here in the US, like how do you deal with the business model across these different sites as you start to cross international boundaries? How do you deal with data privacy law, differing data privacy laws, data residency laws? Another thing we're going to have to confront. And a lot of countries won't let this type of data leave their geography. And so how do you really federate across countries and geographies? I do think something like the architecture I described in my prior comments about kind of one common or one conceptual TRE with kind of footprint in all these countries, and again, kind of standardizing as much as possible is the direction we're headed. And we think the market will respond to that ultimately. That we know large pharma is sourcing data from around the world in that regard. So if we can help bring these data sources together, we think there'll be demand for that.
Yeah, because I guess there's a lot of different software providers and people who build custom trusted research environments, right? And I don't know that there's a common standard that they interoperate on. So do you end up in a world where you have, an oligopoly of different systems, or do you have some kind of layer that allows someone to run the analysis in a federated way, no matter whether they're using, whoever it is that's built the TRE.
Yeah, and that's where, and I mean, I think even large pharma, for the most part, is still using their own. Like a lot of them build their own
Right.
systems internally. They're not adopting that many of the commercial tools. And yes, there are probably 6 or 8 different providers in the commercial tool space at this moment. That was an area where we didn't wish to build our own platform, so we've done it by partnerships, and we'll announce later that this spring kind of what that will look like ultimately. But certainly, I mean, differentiating kind of the tools from how the data is structured in that regard, and that's where like common data model across that's a separate thing from the tooling. And so that's where I think starting to standardize data into model and vocabulary so that it is more interoperable, and then it could flow into different tools in that regard.
Yeah, interesting. As we finish off here, I know you probably have some perspectives, more of a current event discussion here, but it has very long-term implications. In the US right now, we have NIH funding in particular undergoing major shockwaves. I'm curious to get your perspective on what are the immediate effects of that, but also what are some of the second and third order effects if we see major changes that that are happening or proposed to be happening in the next couple of months and years.
Yeah, this is an area that's near and dear to my heart for a number of reasons. One, I did a PhD in biomedical engineering, so I came through kind of an age-funded academic medical center. Functional brain imaging was my area of expertise, which requires a tremendous amount of resource in terms of building imaging resources. My wife is a faculty member at the University of Colorado near where we live, and so I'm kind of acutely aware of what's going on in that regard. But I look back at the reason BioView exists at Vanderbilt is because of the science that was powered off of it. And so thinking about all the things that we've been able to accomplish with NashBio, one of the big motivators for Vanderbilt in building this was to power their own science, which is ultimately funded by, in many ways, the federal government here in the US. And so to see the disruptions to basic science funding is alarming in that regard. Does it affect me right today at NashBio? No, but it certainly will be downstream. And so all of us sitting in kind of that biotech layer, as you kick the legs out from basic science, what's going to happen 3 to 5 years from now, 20 years from now? It seems it's alarming and quite concerning in that regard. That the innovations that we're able to derive don't just come from thin air; they come on the backs of the basic science work that our colleagues in various academic settings are doing. And so the disruption to that is concerning and alarming. And I don't claim that there's not room for efficiency. I mean, certainly there's opportunity to look at the ways the NIH functions and asking which disease areas or which research projects. But as best as we can tell from the outside, it seems like there's throwing sand in the gears of the whole thing just to grind it to a halt. And it's going to have impact. It has impact right now on the scientists working, and it will certainly have impact for those of us in the biotech ecosystem as the discovery is not made or discovery is slowed down in the basic science layers as that shockwave moves through the system, which is biotech is certainly one of the crown jewels of the U.S. economy, and so that comes on the backs of investments that we've made here in the country for 100 years or more in that regard. And so it's as I say, it is quite concerning as somebody who operates in the beneficiary layer of all that basic science work.
Yeah, and I think the numbers are pretty clear that most large pharma biotech innovation comes from biotechs that they're either buying or partnering with, and most of those cases you can ultimately trace back to an academic medical center where someone has invented a new technology or discovered a target through big genetic analysis or something, or invention of a tool. So I think the logic chain is pretty clear that disrupting that base layer is going to have pretty major knock-on effects. What's hard about it is the time lag. I think that you mentioned that you probably don't see something this year, you don't see something next year, but it may be 2-3 years from now we notice a major cliff in innovation, right? Because of the shock to the system at the very early stages that then ripples downstream.
Well, and then ultimately 10 years from now, fewer drugs coming to market, I think is the impact, is the eventual impact, right? Is like there'll be less, as that innovation shockwave moves through, less benefit for patients, unfortunately.
Yeah. Is there a solution to this besides try to keep the same level of science funding, but streamline operationally? Because I think you mentioned before that you don't dispute the fact that we may be over administering some of these projects or potentially funding things in the wrong way, right? That there's a lot of discussion of having participated in grants before. Is that process overly onerous where people are testing funding investigators, right? Go do good science and prosper versus you have to write a major grant application.
And that's, I mean, certainly that, again, having been through been in this area for 20 or 25 years with first myself and then watching my wife progress, the amount of time investigators do spend writing grants, the amount of time it takes to adjudicate those, minimum 9 months, if not 18 months from kind of starting to write something before ultimately comes out. Like you look at that and like there has to be ways we can speed some of that up. Certainly cost potentially comes out of the system or gets routed back into the system. And I guess maybe the philosophical question is like, what's the best way to find that? Is it to do it to the system or to ask the system, how could we make this more efficient? Because I suspect many folks who interact with the NIH would have a number of ideas about ways to make it more efficient in that regard. And so if like bringing people along on that journey as opposed to doing it to them, I think would ultimately lead to, I believe would lead to a more successful outcome in terms of making it more efficient. But yeah, again, it's no question there are ways to make it to improve things. That the timelines and the number of grants that individual investigators are writing to be able to fund the research they're doing, like there is a bunch of wasted effort in that regard. Question: how do you find that wasted effort? How do you design a new system out of it? I would say a more collaborative model, I think, would lead to better outcomes.
Yeah, I am personally interested in putting more money in testing behind models that fund broad direction and investigators that have a vision and idea versus hyper-specific projects. Because, I think we know that in science, often the most interesting ideas are contrarian and they don't make it past a committee, right? That's looking for the next incremental thing. There's pitfalls with that approach. Obviously, you don't want to fund the same people for things that aren't working. But I think that feels like a direction that cuts down on some of the administration as well, of having to have huge adjudication committees. There have been some interesting, I think, philanthropic attempts to do this. And in the UK, like if I think about the Wellcome Trust, for example, they tend to do more core funding of institutes for 5 years and give people a little bit of a longer horizon versus, it's a really tightly controlled 2-year grant or whatever it is. So I think there's an opportunity to innovate. But yeah, I'm watching with concern around what the third order effects of this change are.
Yep, I would agree.
As we wrap up here, what's the most exciting thing that you're working on for the next couple of years? You're going to close out this first 250,000 people. You mentioned you've got 100,000 or so more that you could start to include, but you're also thinking about proteomics, RNA sequencing. Where if you were to name the top 2 or 3 things you're focused on from here, what would they be?
Yeah, I think it's the overall continued growth of NashBio. One of our missions since the beginning is how to make this data more easily accessible and useful to the companies we work with. And so some of the innovation around delivery model and business model that we're working through, we're quite excited about that. We're actively discussing with our partners in the alliance about what should be the next directions to go. Is there interest in more data generation, whether it's adding additional genomics or adding other omics layers? Obviously, we saw the big UK Biobank announcements in early January about the kind of expanding to the whole cohort. So we're definitely picking up an interest in proteomics in that regard. That said, the thing we're trying to think through is like, are there other things that we could do? Like, should we just follow the UK Biobank from that standpoint, or are there things that we could uniquely do that would continue to set BioView and NashBio apart from some of these other resources, or complement them? I think is probably maybe the better way to say it.
Yeah.
No single resource of this type there's no one-stop shop in that regard. And so thinking about, is there more disease-specific context? Some of the advantages we have with the biobank is we can do serial sampling over time of how is the blood proteome evolving as somebody is moving through a disease or a treatment. That's one idea we've been discussing with the various members, looking at tissue samples from residual disease. So being able to do, whether it's bulk transcriptomics, or could you open up a metabolomics layer or something like that? I think we're kind of all options on the table. I would bet proteomics will probably be part of it. Just that pharma's spoken pretty loud and clear, but some of those other areas that allow kind of further understanding of disease mechanism and ultimately biological pathways. And again, what can we uniquely add to the scientific data ecosystem? That'll certainly be an ongoing discussion. And in some sense, the AGD first round benefited from a very simple idea. We're just going to go sequence as many samples as we can. We are at a spot where there's many different possibilities. And so sorting through which ones are going to be most useful, and there's many degrees of freedom right now. And so that's where it's fun. It's also challenging. How do you kind of coalesce and build support around 1 or 2 ideas when there's a lot of different directions you could go? So we'll come back in a few years and tell what the result of that story was. But I learned a ton with the first AGD. We'll see if we can put it to practice in further extensions of it.
Amazing. Well, congratulations on all the great progress. Thanks for sharing with us today. If people want to follow you, follow your work, the website is nashbio.com. So people can take a look there. Are you looking for anything in particular? Partners, hiring? How could people be helpful to you?
Yeah, we are always open for business in that regard. And I say particularly as we move to the more all you can eat or self-serve model. We're looking for companies to take advantage of that and help us figure out what exactly that model needs to be. And then certainly looking at therapeutic area, what areas of therapeutic specialty are most interesting to the broader biotech ecosystem at this point. But cardiometabolic and obesity is certainly, don't need to follow the news that closely to posit that's an area of interest. But what else does pharma demand from data providers like us over the next few years? So certainly like to listen to that feedback as we think about building out our data product ecosystem to better serve the industry.
Amazing. Well, thanks again for being with us. And thanks everybody for tuning in. And as always, we'll see you next time.
Great. Thanks, Patrick.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.